Welcome everyone to Students for Science videos on biology, chemistry, physics, math, basically all the sciences. My name is Elias Vanderloon and I will be introducing you to the biological and medical related material. And this is my first video. It's on lipids and membranes. I know most of you guys are probably tired because of studying on hours and going through YouTube videos, so just listen to my Moroccan voice. Maybe you can throw in some spice in your education and bring in some sensuality into your learning. No, but in all seriousness, let's get started. Let's look at the phospholipid bilayer. Now, most of you should be familiar with this. Essentially, we have our hydrophobic tails on the inside, and our polar head groups that are hydrophilic. These are basically the phosphates, and these are going to be negatively charged. Now, this is a sheet-like structure that are two molecules thick, as you can see, 60 to 100 angstroms total. And the phospholipid bilayer consists of lipids as well as proteins, and these proteins each serve different functions, whether it's pumps, channels, receptors, enzymes, etc. And the whole thing is kept together through a series of non-covalent forces. Now, think back into Gen Chem. Remember, we have van der Waals forces, the hydrophobic interactions, we have hydrogen bonds that we will see on either side of the membrane, as well as ionic and electrostatic interactions. Now, the entire membrane is very asymmetric in that surfaces are completely different from each other, and it's also very fluid. And because it's fluid, it's often referred to as a mosaic structure. And what is meant by a mosaic structure is that the phospholipid bilayer is fluid enough to allow things to pass through it and allow for flexibility of the cell, but rigid enough to maintain the constants within the cell. It also allows for the electrical polarization of the cell, and this is extremely important when we go ahead and talk about, say, neurons and, I don't know, maybe action potentials. But this is an oversimplification of the phospholipid bilayer. Don't ever look at this again. Here we go. This is a real one. Or, not a real one, but it's a lot prettier at least. So, you see, phospholipid bilayer is complex in that it has so many different proteins and phospholipids all over. We have our glycolipids right over here that are generally used for signaling. We have integral proteins, such as this globular protein that spans the membrane. Generally, we'll see associated proteins on either side of the membrane that play particular roles. We'll see integral proteins that are associated with one leaflet of the phospholipid bilayer. We'll see pumps, we'll see channels, we'll see enzymes, but we'll talk about proteins another time. Today we're going to talk about lipids. And if you look at the phospholipid bilayer, well, it's made of a whole bunch of phospholipids. And these phospholipids contain fatty acids right over there, and we also have cholesterol. And so, since the phospholipid bilayer is made up of so many fatty acids, well, that's where our conversation is going to start. So, let's go ahead and get started at that. Here are some basic fatty acids. So, look at arachidic, stearic, and palmitic acid. And the first thing that should hit you is that they're all straight. But let's talk about the general structure first, I guess. So let's, if we look at the polar head, this is basically the phosphate, right? And it's negatively charged. And then we have two fatty acids. And this is one fatty acid. But we have the polar head and one fatty acid. And this is going to be apparent for all of the fatty acids we're looking at. But the reason why these are straight and these are not is that these are saturated fatty acids. And what it means by saturated fatty acids is that there are no double bonds. So if we look at something like oleic acid, we see that kink. And that kink is due to a double bond. It is not due to just any double bond. It is a double bond in the cis configuration. Now, biologically speaking, most fatty acids we're going to be looking at are going to have double bonds in the cis configuration, not in the trans configuration, because, well, quite frankly, trans configurations generally are natural. Now, the reasons why we've heard so many bad things about trans fats on the news and wherever else is because they do not display the same biological characteristics as double bonds in cis configuration. So let's go ahead and look at oleic and eleatic acid. And here we go. If you look at the double bond in the oleic acid that's in the cis formation, we see a kink. That's a kink. This is not a kink. Alright? And because the kink is not there, you know, the membrane doesn't get kinky enough, and it's just overall bad. But seriously, the reason for this is because in order for the membrane to maintain fluidity, it requires flexibility. This adds to the flexibility, but when we're going to start talking about melting points, we're going to see that the more double bonds there are in a fatty acid, the lower the melting point. And the lower the melting point is something, the more fluid it is. And so if all of the double bonds that are inserted into, all of the fatty acids with double bonds that are inserted into the membrane for the sake of fluidity are in the trans formation, in trans configuration, then we're not getting a fluid plasma membrane. And instead, we're getting something that's so much more rigid than that. So that's food for thought. We'll return to the melting points at a later point. But let's look at nomenclature really quickly, then we're going to look at essential fatty acids, and then we're going to talk about specific types of phospholipids. So if we look at palmitate and oleate, and the reason why they're called reason why they're phrased this way is because the ionized form of palmitic acid, and that basically means that the carboxyl group donated the hydrogen. If we try and count the carbons, starting from the carboxyl group, as we remember from organic chemistry, it's going to be 1, 2, 3, 4, 5, 6, 7, 8, 9. Since this is cis, this is going to be cis delta 9 oleate. Alright? Nice, simple. But there's another way we can count these carbons, and that's using the omega counting system. Now, to count via this system, we're going to start with the carbon that is the methyl group at the very end. And so this is going to be first one, second omega carbon, and here we go, the third omega carbon. And if you look at that, this is where the double bond lands, and so this is going to be an omega-3 fatty acid. I'm sure you guys have heard of omega-3 fatty acids and their health benefits. So, anyways, I want to keep this video under 15 minutes. I want to get through everything, so just going to keep moving forward. Let's talk about essential fatty acids. So right here we're looking at methyllinolinate, and the reason why it's an essential fatty acid is because it has 3 double bonds beyond the 8th position, beyond the 8th delta position. Naturally, us as animals cannot make these double bonds beyond the 8th position within our own biological systems, and because we can't, it's an essential nutrient. Alright, so now let's ask the question, since it has 3 double bonds, is it going to have a higher melting point or a lower melting point compared to something with say 2 double bonds? And the answer is it's going to have a lower melting point. So to phrase this right, the higher the degree of saturation, the higher the melting temperature is. So meaning the less double bonds that are in the fatty acid, then the higher the melting temperature will be. And it should go without saying that the longer the chain length, the higher the melting temperature will be. So before we get into the different types of lipids, I just want to remind you all that these lipids and fatty acids aren't just for the membrane, right? I mean, they're fuel molecules, they're highly concentrated energy stores, they're often used in signal transduction, and of course, well, you know, we see them in our plasma membrane, and that's really important. So now for the different types of lipids, we have phosphoglycerides as the first one we're going to go after. Now, these are amphiphilic, meaning that they are hydrophilic at one end, hydrophobic at the other. Why are they phosphoglycerides? Well, glycerol is the backbone. Alright? And just to keep this in mind, once a molecule is added to the phosphate, this is a phosphatide. So let's look at an example, and you guys should probably be familiar with the structure of glycerol, so we're going to look at that as well. Alright, it's too big. Here we go. So our phosphoglyceride, we have the hydrophobic tail. Here's our glycerol. Notice how the fatty acids are actually bonded to oxygen. That's if you remember, this is the carboxyl group right there, and this is therefore the delta carbon, and then the other end is going to be the omega carbon. But look at all those O's. So my little nickname for glycerol is the O-man, and kind of like Santa Claus for organic chemistry, if you know what I mean. No? Maybe? Okay. Well, anyways, that's phosphoglycerides. We're going to move on really quickly, and we're going to go right into the next type, and that is sphingophospholipid. And instead of glycerol, they have sphingosine as their backbone. So it's often found in the brain. One unusual sphingophospholipid is sphingomyelin, and it's unusual because, well, it has a phosphate on it, and by its definition, well, it's in myelin. It's found in myelin sheaths in the brain. Now, sphingosine I find pretty interesting because it's freaking big. One monster right there, compared to glycerol. And you know, they say size doesn't matter. Alright, moving on, we've got my favorite piece, glycolipids. Why are they my favorite? Because they're glycosylated, they're sweet, sugar. Sugar is pretty sexy if you ask me. Anyway, so let's look at the basic structure of a glycolipid is essentially a lipid attached to a sugar, and it contains a sphingosine moiety with a fatty acid attached to the amino group of one or more sugars, and the sugars are esterified to the primary hydroxyl group right there. If you look at ganglioside, well, this is not just some simple sugar. This is an oligosaccharide. One interesting fact about ganglioside is that it's found to be important in the growth and differentiation of tissues, but it's also found to be important in carcinogenesis, and it has been found that tumor formation can induce the synthesis of a new complement of ganglioside, and very low concentrations of a specific ganglioside can induce differentiation of cultured neuronal tumor cells. One other interesting fact is that if you get rid of the oligosaccharide right there, yep, chop that off, and you have ceramide. Ceramide has an apoptosis-inducing effect on cancer. So, if you think cancer is something you're interested in, and it's biochemistry, go look up those two fatty acids. Moving on. Steroids. Your mom always say not to do these. Well, okay, they're not steroids. It's cholesterol, and it has a steroid scaffold. So this is the fourth type of lipid we're going to be looking at today. And just sorry for the bad resolution here, but I'm trying to go through this with you. We have the hydroxyl group on this one end, and this is the esterification site, one bound to something else, and then the hydrocarbon tail on the other. Now, unlike the other lipids we're looking at, this is not amphiphilic. Okay, it mostly resides in the hydrophobic region of the membrane. Pay attention to the hydroxide on the corner because that is the only hydrophilic part of the molecule, and it's an important component of the membrane. So think about it. If it's hydrophilic, it'd probably be closer to the head of the fatty acid. So it'd be trying to get outside of the membrane. If anything, perhaps even anchored into that part. Steroids are great for the membrane because they increase rigidity, and that's extremely important when we're talking about the mosaic structure of the phospholipid bilayer. So for part one of this video, I am just going to leave you with this beautiful photo of the phospholipid bilayer. So we have our glycolipids there, our cytoskeleton down there. This is a different photo. But our cholesterol here essentially acts super rigid. So see you in the next video.